Hello and welcome back to Mr. Carter Science Specials. Today we'll be looking at the Edexcel IGCSE Biology and Double Science course, and we're still looking at the B2 Structure and Function of Living Things. This particular video will be focusing on B2A, the movement in and out of cells, movement of substances in and out of cells. As always, I offer you the following advice. Please listen carefully, pause the video often, make notes using the slides, and complete the review questions before listening to the answers. See what you can learn. This is all about learning. As always, we have the syllabus references there, so you can follow along with the course and the syllabus at your own pace. So, little introduction. There are only three ways that a substance can move in and out of cells, just three: diffusion, osmosis and active transport. If you ever come across an exam question which is asking you explain how or state how a substance can move in and out of a cell, you've got a choice of three words. Pick one. It's a 1 in 3 chance. You never know, you might get it right. But let's hope you can, after watching this video, you'll be better able to get the correct answers. So the first way that substances can move in and out of cells is called diffusion. And diffusion is the movement of a substance from a high concentration to a low concentration. So if we'll have a look at this little diagram here, we've got somebody dropping some dye molecules into a beaker of water, and initially we have a very high concentration of those dye molecules here, but away from that area a very low concentration of those dye molecules. And over time, because the particles are moving, because it's a liquid, and in a liquid particles are always moving in a random direction, just by random movement this high concentration of particles will begin to move, diffuse and spread out to the areas of low concentration. Now in biology we always talk about areas of high concentration and low concentration because we can measure concentrations, so we can predict the movement and which way substances will move either in or out of a cell. The most important thing to know about diffusion is that it is passive, so it doesn't need any energy at all. So when diffusion happens in your body, you don't have to waste any energy trying to make molecules move in a certain direction. So in diffusion, the molecules will always diffuse until we reach an equilibrium like we have here. And an equilibrium is when we have equal concentrations of our particles, in particular on the inside and on the outside of cells, or in this example, spread throughout the beaker. So here is another example of diffusion with the high concentration spreading out to the low concentration, and at this point we're beginning to reach equilibrium. So what kind of substances can move in and out of a cell by diffusion? Well, they all have to be substances that can move through the cell membrane and dissolve through the cell membrane. We call these substances lipid soluble because the cell membrane is made out of lipids. So things like oxygen, carbon dioxide, urea and glucose can all enter and leave a cell by diffusion. Glucose, it should be said, needs an extra little bit of help. It needs some special proteins with protein channels, and that's a special kind of diffusion called facilitated diffusion. So let's look. So what kind of factors will affect the rates of diffusion that are happening in your body? Well, things like the temperature, concentration, the surface area to volume ratio and the distance. These will all have an effect on the rate of diffusion that's happening inside your body. Now remember, a rate is the amount of diffusion per time, often per minute or per second. So here we've got some villi which are found in the small intestine, and these villi are very well adapted to take into account all of these different factors. So before we move on to the next slide, try this task. Explain how each of the factors will affect the rate of diffusion. Pause the video and we'll give you the answers in a moment. So let's have a look at these factors one by one. Temperature. If we were to investigate how temperature would affect the rate of diffusion, we should end up with a graph that looks like this. And we would say, as the temperature increases, the rate of diffusion increases. It's a positive correlation. A higher temperature means that those particles are moving much faster, and if the particles can move faster, then they'll spread out faster, so diffusion will happen quicker. If we look at concentration, diffusion is the movement of a substance from a high concentration to a low concentration. So the bigger the difference in those two concentrations, the faster the rate of diffusion will happen. So if we have a very high concentration to start off with, we will end up with a very large rate of diffusion. Surface area to volume ratio. Again, the greater your surface area of your cells, the more surface you've got, the more diffusion can move through your cell membranes. So a larger surface area compared to your volume will increase the rate of diffusion. As surface area to volume ratio increases, the rate of diffusion will increase. And then finally, distance. We really want to have a very small distance for diffusion to happen over in the human body. That way diffusion will happen very quickly if we're only moving from perhaps 1 millimeter to another millimeter. If we're happening this over millimeters or even micrometers, then we're going to have an incredibly fast rate of diffusion. So actually we've got a negative correlation here, where as the distance increases, the rate of diffusion will actually decrease. We want to have a very short distance to have a fast rate of diffusion. So as I've said, you've got a large number of organs which are very well adapted to make certain that they have diffusion happening in the fastest possible ways. You need to know about 3 of these, the first of which is the small intestine. So if we have a quick look at this diagram, here we've got a large tube, and inside this tube here it's full of villi, but it's also full of enzymes and your food where digestion is occurring. And all of those food molecules, once they've been broken down into the smaller molecules, they want to be able to move through here and into the blood. So if you have a look at that in a zoomed in fashion, we have here all of that blood, and we want to try and get as much of the food molecules into there as quickly as possible. So how does this work? Well, quite obviously there is a high concentration of nutrients in the small intestine and a low concentration of nutrients in the blood because you've just eaten a meal. So the nutrients will diffuse from the small intestine into the blood. And the small intestine has many adaptations to help it absorb food faster. Firstly, it's got villi and then microvilli, which if we have a look over here, here is a diagram of one villi, and if you look at each individual cell, and they're zoomed in here, we can see each individual cell is full of little folds of a cell membrane called microvilli. So the villi and the microvilli give the small intestines a very large surface area, and as we've just seen, this speeds up the rate of diffusion and allows nutrients to be absorbed into the blood faster. The walls of the villi are also 1 cell thick, and this provides a short diffusion distance. So between the small intestine and the blood, so diffusion of the nutrients is therefore faster. Each villus, many villi, one villus, each villus also contains a lacteal to help absorb glycerol and fatty acids, and the capillaries provide a flow of blood, a good blood flow, and that helps to maintain the concentration gradients. So every time it looks like we're going to reach equilibrium, the blood flows away, removing all of those nutrients that have already been absorbed. Therefore, we're constantly moving from a high concentration in the small intestine to a low concentration in the capillaries in the blood. Every time those concentrations get closer, the capillaries just move the blood away and replace them with a low concentration. That maintains the diffusion gradient. So the next organ we're going to look at is the placenta. The placenta is an organ which is used in pregnancy to supply the growing fetus with nutrients and also to remove waste products. In the placenta we have the mother's blood and the fetus's blood, which come very close together, but they do not touch. It's a very short distance, and that allows the oxygen and nutrients to diffuse from the mother's blood into the fetus's blood, and also waste products like carbon dioxide and urea to diffuse in the opposite direction. Also, substances like antibodies, minerals and amino acids can also diffuse across the placenta and protect the fetus and help it grow. Unfortunately, harmful substances like nicotine and alcohol and other illegal drugs can also cross the placenta, and these can harm the growing fetus. So what kind of adaptations does the placenta have? Well, the placenta has lots of different adaptations to help speed up diffusion. Firstly, if we have a look on this diagram here, we have sections of the fetus's blood which are looking very much like villi. And then if we have a look at the mother's blood as well, the mother has villi working in the opposite direction. And these villi provide a very large surface area to ensure fast diffusion. The villi also have very thin walls to ensure a short diffusion distance, which will also mean a fast rate of diffusion. And there's also a large blood supply bringing in all the nutrients and creating a big diffusion gradient, so again diffusion is faster. So if we have a look at the alveoli in the lungs, these are also an organ where we need to have a large amount of diffusion. And if you have a look here, we have the blood flowing in and it's carrying lots of carbon dioxide, and as it leaves it's carrying lots of oxygen, and the carbon dioxide will diffuse out of the blood and the oxygen will diffuse into the blood. So what kind of adaptations does the alveoli have? Well, you can pause the video now and try this gap fill. So here are the answers. Gas exchange takes place in the alveoli, and the alveoli have a very large surface area for gas exchange to take place. The alveoli walls are very thin, as are the capillary walls, so there's a very short distance for diffusion. So diffusion can take place very, very quickly. The alveoli also have a good blood supply to maintain that diffusion gradient. Now you may have noticed over these last three examples that we have the same phrases coming up again and again and again. We need a good blood supply to maintain the diffusion gradient. We need to have a very thin wall so that there's a short diffusion distance. And we need to have a large surface area so that we can have very fast diffusion. And if you can mention those three things for the three organs that we've mentioned, you should pick up 3 marks in your exams. So pause the video now, try these review questions all about diffusion. So let's have a look at some of the answers then. Define the term diffusion. Well, diffusion is the movement of a substance from a high concentration going to a low concentration. Give examples of substances that can diffuse in and out of a cell. Well, we could talk about oxygen, we can talk about carbon dioxide, and we can talk about urea. We could also mention glucose, but only if we have those special proteins for facilitated diffusion. Name 4 factors that affect the rate of diffusion and explain how they affect the rate of diffusion. So our 4 factors, we had temperature, and there's a special sentence we're going to try and use here now. As temperature increases, the rate of diffusion increases. I'll say that sentence again. As temperature increases, the rate of diffusion increases. This is a very important sentence. It always starts with as the. And if you can remember this sentence, anytime you're trying to describe a graph or describe what happens when we're looking at different factors affecting something, then you should do very well with those questions. As the temperature increases, the rate of diffusion increases. The second factor we were looking at was the concentration. And it's the same sentence. As the concentration increases, the rate of diffusion increases. The third factor was the surface area compared to the volume, surface area to volume ratio. As the surface area to volume ratio increases, the rate of diffusion increases. And then our final one was the diffusion distance. And as the distance increases, the rate of diffusion decreases. We really want to have a short diffusion distance to have fast rate of diffusion. Question 4. Explain how the small intestine is adapted for fast diffusion. Well, we need to make certain that we talk about having a large surface area. We need to talk about having good blood supply to maintain the diffusion gradient. We need to talk about having thin walls so that we have a short distance for diffusion. And that's the same for question 5 and 6. In the placenta, we have villi, which provide a large surface area. We have a good blood supply to maintain the diffusion gradient, and thin walls so we have a short diffusion distance. In the alveoli for question 6, we have a large surface area, so we have a fast rate of diffusion. We have a good blood supply to maintain the diffusion gradient, and thin walls so we have a short distance for diffusion to happen. It's the same 3 points, and it doesn't matter which organ we are looking at. We can still pick up at least 3 marks on how they are adapted. So the next way that a substance may be able to move in and out of a cell is called osmosis. So if we have a look at this diagram, we can see what is happening in terms of osmosis. And the first thing we need to notice is that we have, number 1, hexagons of glucose and round blue circles of water. We also have a semi-permeable membrane dividing the page. So if we consider that this is the inside of a cell, and that this might be the outside of a cell, we can begin to understand what's happening and why. If we consider the glucose first of all, on this side we only have 1 glucose molecule, so that would be a low concentration of glucose. However, outside the cell we have a very high concentration of glucose. So you would expect that the glucose would try and move from high to low by diffusion. But if you look carefully, 1 glucose molecule cannot possibly fit through the gap in the semi-permeable membrane. Therefore, we cannot possibly have any diffusion of glucose. So how else can we try and make the concentrations both inside and outside the cell equal? Well, one of the ways that we can try and make the concentrations equal is to consider the movement of water. If this side gains water, it will become more and more and more dilute, and the concentration of water will become less and less and less inside the cell. But if the inside of a cell loses water, and those water molecules begin to move through that semi-permeable membrane just randomly, because all water molecules are moving in a random direction. Some will move up, some will move down, and some might move towards the cell membrane. Then the inside will lose some water molecules, and therefore the concentration inside the cell will increase. And if the outside of a cell is gaining water molecules, it's going to become more and more dilute. That glucose will become less and less concentrated, and the concentration of glucose will decrease. And that's in fact what happens when we cannot move the glucose due to the semi-permeable membrane. The water will move in the opposite direction. We will go from a high concentration of water to the low concentration of water. But it's really hard to measure water concentrations, but it's easy to measure glucose concentrations. So when we describe osmosis, we talk about movement from a low concentration of glucose or a low concentration of our solute. whatever is dissolved in the solution. So water, osmosis, is the movement of water from a low concentration of solute to a high concentration of the solute. Low to high. This is the opposite way to diffusion, but we can only have osmosis when we have a semi-permeable membrane. So here's our full definition of osmosis. The movement of water from a low concentration to a high concentration through a semi-permeable membrane. Osmosis works in the opposite direction to diffusion, and osmosis is passive. It does not need any energy at all. So if we have a look here at a number of different cells, if you pause the video now, you can begin to try and work out what is happening to each cell, where is the water moving and why, and you might even be able to work out where the different concentrations of solutes are. Where is the high concentration? Where is the low concentration? So I'll give you one example before I talk about all four. If we have a look at this cell here is an animal cell, and it's undergoing a process called lysis. Lysis happens when an animal cell bursts. The reason this cell has burst is because it's had far too much water moving in. Well, as soon as we know that the water has moved in, we know exactly where all the concentrations are. We can use our definition of osmosis. So osmosis is the movement of water from a low concentration to a high concentration through a semi-permeable membrane, low to high. So straight away we know the outside of a cell had a very low concentration and the inside of a cell had a high concentration, and it drew in the water. We can look at the exact opposite scenario here, where we have a red blood cell which has crenated, and that is because it has lost water. And again, we can use our definition. Osmosis is the movement of water from a low concentration to a high concentration. I like to do high concentrations in red. So in this environment, that red blood cell has been placed in a concentration which is incredibly salty or sugary, a very high concentration. Therefore, it's lost water, and we say it has been crenated. Incidentally, the lowest possible concentration of water you could have is pure water. Pure water is the very lowest concentration of water we can have because it has absolutely nothing dissolved in it. So if we place a cell in our pure water, then we will always start to see lysis of our cells in animal cells. However, plant cells have a very strong cell wall surrounding them, and this protects them. So in this particular case here, when we look at turgidity, this cell is very fat. It's got a lot of water inside it because there's a lot of water moving in. So again, we're going from low concentrations outside the cell, possibly pure water, to a high concentration inside the cell. But we don't get lysis because the cell wall protects the cell membrane from breaking, so the cell will not split. This incidentally is why leaves will often be able to stand upright and point out, because all the cells are very turgid and all pushing against each other. But we also have this cell down here where we have a plasmolysis happening. Now in plasmolysis we've got the exact same thing we have in crenation, in that the cell membrane, and I'll just mark the cell membrane on so we can see it clearly. Here is the cell membrane of this cell, and it's pulled away from the cell wall. Why? Because we've had a large amount of water leaving this cell, because the cell has been placed in a high concentration, and in the cytoplasm inside the cell we have a low concentration. So the cell will lose water, but it doesn't change shape much because we still have the cell wall there holding the cell in its normal shape. However, the cell wall is fully permeable. So this high concentration solution will actually flow in through the cell wall, and it will fill all of the spaces between the cell wall and the cytoplasm. I have seen an exam question which was asking about this, and a number of students thought that would fill with air. But that's, if you think about it, it's an impossibility. If we place these cells into a particular solution, that solution will flow through the cell wall because it's fully permeable. Anything can move in and out of it. Now, of course, you might not like the scribbly notes that I put on here. So on the next slide, we have slightly better notes for you. Again, I would recommend perhaps that you print this slide off so that you can understand exactly what's happening here. So the final way is active transport. The final way that a substance can move in and out of a cell is active transport. And active transport is the movement of a substance from a low concentration to a high concentration. Again, very much like we had in osmosis, we're moving from low to high. We're going against the concentration gradient. But active transport is active. That means it needs lots and lots and lots of energy. Osmosis and diffusion are passive, they don't require energy, but active transport needs lots and lots of energy. And that's because it uses a transport protein in the cell membrane. So let me explain what's happening. You can see it in this diagram here. If you have your solute moving into this transport protein, so it's moving in like this, if you add some energy, so if our solute comes in, and then you add some energy, the protein can change shape, and the solute will move through the protein, and it's now inside the cell. And the transport protein will then reset itself, ready for the next solute to move in, so that it can again change shape, and that solute will move inside the cell. So there's a lot of different chemicals that can only enter and leave a cell by active transport. Sometimes, for example, potassium ions here, sodium ions, amino acids, they all need active transport. And then glucose. Although glucose can move into the cell by diffusion, it will often have active transport as well, because the cell will want to try and have the highest concentration of glucose it can possibly get, so that it can carry out a large amount of respiration. So here we have an example of how diffusion and active transport can work together in the small intestine. As we've said previously, in the small intestine here, there is a high concentration of nutrients and a low concentration of nutrients in the blood in the villi. So the nutrients will diffuse from the small intestine into the blood. But diffusion stops when the concentrations reach equilibrium, when they're equal. And this would leave some nutrients in the small intestine, which means when you go to the toilet, you'd be losing some of the nutrients that you've spent your energy trying to consume and eat. So when the diffusion reaches the equilibrium, active transport is used to absorb the remaining nutrients. So during digestion, the small intestine absorbs the soluble nutrients, but over time they reach the equilibrium. So active transport is used to continue the transport of the small amounts of remaining nutrients against their concentration gradient. And the cells of the villi have a large amount of mitochondria, which will provide the energy for those transport proteins to change their shape and move and allow active transport to happen. So in these villi cells here, and all the cells that are lining the villi here, we have a large amount of mitochondria providing the energy through respiration. So a very useful adaptation of the small intestine to allow active transport to occur. So here's an interesting question. Explain how plant roots absorb minerals and water using both active transport and osmosis. Just pause the video now, write that question down. It would be a 6-mark question in an exam. See whether you can do it. If you can't, on the next slide there'll be a gap fill to help you, and then we'll go through the answers on the 3rd slide. So here is the gap fill if you need some additional help. And finally, here are the answers. Explain how a plant root absorbs minerals and water using both active transport and osmosis. Well, minerals like nitrates and magnesium are in a very low concentration in the soil. So if we can add that onto our diagram here, they're in a low concentration in the soil. But they're in a very high concentration in the root hair cells. So the minerals move from a low to a high concentration by active transport and they enter the cells. This of course needs energy, and the energy is provided by mitochondria in the root hair cells. The concentration in the root hair cells increases, and there is now a low concentration outside the cell and a higher concentration in the cell. So this also allows water to enter by osmosis. So water will also flow because active transport has created the concentration gradient that will allow water to flow into the root hair cells. So the root hair cells, by using active transport to absorb minerals, also create concentration gradients that allow water to flow in by osmosis, and that's how plants are able to suck up water. It's also quite interesting how and why, if you overwater a plant, if you've got a plant and you keep putting water on the soil, keep watering it, it turns out you're not doing the right thing for the plant. Because what you will often do is you will fill up all the different little air spaces in the soil. And if you completely flood those air spaces with water, there's no oxygen for the root hair cells to use. And if there's no oxygen, the root hair cells can't do respiration, they can't produce energy, and there's no energy for active transport, so they can't draw in the minerals. They can't create the correct concentration gradient for osmosis to happen. Therefore, the plants won't be able to absorb minerals or water, and the plant will eventually die, even though you're providing lots and lots of water for it. So please don't overwater your plants. So here's a number of review questions about osmosis and active transport. Pause the video, try these questions, and we'll go through the answers next. So, define the term osmosis. Osmosis is the movement of water from a low concentration to a high concentration through a semi-permeable membrane. Define the term active transport. Active transport is the movement of a substance from a low concentration to a high concentration using active transport or transport proteins, and active transport needs energy. Give examples of osmosis in living organisms. There's lots of different examples you could use here, but you may go with the root hair cells absorbing water and water moving from cell to cell to cell in a plant using osmosis. You may choose to go with osmosis being used in the kidney in humans to reabsorb water so you don't lose too much water if you're dehydrated. Give examples of active transport in living organisms. Well, there's a range of examples you could use here. You could use cells absorbing potassium ions or calcium ions or sodium ions by active transport. You could go with root hair cells absorbing a variety of minerals or magnesium by active transport. And then question 5, compare and contrast diffusion, osmosis and active transport. So you could talk about diffusion being the movement of any substance from a high concentration to low, and it is passive. Osmosis is the movement of a substance of water from low to high, so it is only water. But again, it is passive. And active transport is the movement of any substance from low to high, but it is active and it needs energy. Thank you for watching this video. I hope it's been useful. I hope you've learned some things. Please make certain that you subscribe to the page and please add any comments below. Thank you.